All right, we will get started for the next section. So this section is the novel cardiac bioengineering intervention.
So our first speaker will be Dr. John Cooke. He is Joseph Walter and Carol Walter Luke Presentation Distinguished Chair in Cardiovascular Disease Research in the Department of Cardiovascular Science. He is also director for the Center for Cardiovascular Regeneration in the Will Clare Medical College. Dr. Cooke. His talk about the title is RNA Therapeutics.
Thank you very much, Mr. Chairman. Thank you all for coming back from the break. I want to thank Dr. Fukuda and Dr. Zhang for organizing such an amazing meeting. It's been really incredible. I've learned a lot. So happy to be here. And I hope we can duplicate this in Houston next year. It's going to be a tall task to do that. You've put on such a great meeting. Well, today I'm going to be talking about RNA therapeutics and how RNA changes cardiovascular drug development and delivery. We're really entering a new era of cardiovascular drug development. And my disclosures, I'm an inventor on RNA technologies assigned to Stanford and to Houston Methodist Hospital, co-founder of a couple of companies related to RNA therapeutics, and on some scientific advisory boards, as you see there. Well, there's a diversity of RNA species, and as you know, and we heard a little bit about the small RNAs yesterday, so I won't belabor that. I'm going to be talking about messenger RNA and a little bit about circular RNA today. There is also a diversity of RNA therapeutics because of the opportunities to manipulate the small RNA and larger RNA molecules. There's a number of companies that have sprung up. I'm going to focus more on the messenger RNA companies and a little bit about the circular RNA companies. There's a lot of excitement around them. In the right-hand lower corner there, Laurent, Orna, Orbital, and Renegade have gotten several $100 million of VC funding each because I do believe that this is, and a lot of other people do, believe that this will be the second generation of long RNA therapeutics, protein encoding therapeutics. The major company in messenger RNA, of course, as you probably know, is Moderna. Their pipeline is already extensive. This company was formed in 2010. They already have over 50 products that are in development, and some that have been approved, as you know, the COVID vaccines. In blue there, you can see that most of their products are vaccines, infectious disease vaccine and cancer vaccines, and that's no accident because messenger RNA lends itself very well for transient expression of an antigen, not so well for protein replacement therapies. That's been a problem for RNA, but that problem is going to be solved, as I'll speak about later. The only product that Moderna has right now for cardiovascular that's undergoing clinical trials is the VEGF mRNA, and here they directly inject, at the time of coronary bypass grafting, messenger RNA encoding VEGF-A, 30 injections around the ischemic zone, and that study is a small study that's been reported out, as Victor mentioned yesterday, with positive trends for left ventricular ejection fraction and BNP and treadmill exercise walking time. So some promising data, more to come. I want you to know about what we've created in the Texas Medical Center for anyone with an RNA idea, and specifically messenger RNA or self-amplifying RNA, circular RNA. We make protein encoding RNAs. We have been helping since 2015, funded by the state government, Texas state government, and NIH. We've been making RNA for folks with our RNA core. This actually, the seed for this, the Anlaga was from PCBC. When we got Victor and Dennis Buxton had the foresight to give us a little bit of funding to make messenger RNA for reprogramming. we started with the Yamanaka factors, and that worked, and we started making RNA for folks in the consortium. When I got to Texas in 2013, I saw this CPRIT possibility. CPRIT was giving out core awards, $1 million a year, and we got one of those to set up an RNA core for cancer investigators. But what we've been able to establish is a team that can help you with your RNA idea, can help you design the RNA construct. We can make it, we can lyophilize it, we can validate it with our GLP assays, we can encapsulate it. We've got a nanomedicine team that works with us. And we have a GMP production. We have clean rooms that are an GMP program overseen by Dr. Chris Lincoln, who we recruited from the City of Hope. He built their GMP facility from the ground up. We have a wonderful GLP preclinical, and we have a first-in-man phase 1. Our problem initially was we could only make small batches, so we teamed up with VGXI. They licensed our technology for synthesis and purification, validation, and they now scaled that up and are ready to do large batches of RNA. So we've got basically a roadmap that we've created all the way to the clinic. Since we began, we've created over 300 RNA constructs for academia and biotech. We've provided over 80 analytical services to support GLP studies and FDA INDs. And as I said, we scaled up the processes for large batch manufacturing. So you can get, if you have a great RNA idea and you have the funding, you can get all the way to the clinic. We're happy to help you. But if you wait 5 years, you won't need us. You won't need us because this whole field is going to change. RNA is like biological software, as you know. Well, the hardware is coming to allow every hospital in the country to have a box on their desk, basically. GMP in a box. Well, you don't need a clean room. The idea will be that you can do personalized RNA therapeutics in your hospital. And this is one device that we're making with Nature's Toolbox, funded by a BARDA grant. Basically, BARDA wanted deployable manufacturing units that they could ship off to low-income countries or theaters of war where it's needed, RNA vaccines are needed acutely. Rather than shipping the RNA vaccine, just ship the equipment and make it in situ. This particular device is a hollow fiber bioreactor system. The reaction enzymes go in at one end with a static template, basically your DNA template on beads. The reaction substrates go in, and it's basically a flow-through system. At the end, you get your RNA with the enzymes, and that's purified because the enzymes are tagged, and we can pull those out, and you get the pure sterile RNA at the end. And what we're working with them is to refine this system and ultimately to include encapsulation with that as well, so you can actually have an RNA drug produced. There's other companies that are in this space. Nutcracker Therapeutics, Quantum, CureVac Printer are the other companies that are doing this. But there'll be boxes in every hospital 5 years from now, and we'll be making personalized RNA therapeutics for, firstly with cancer. I'll show you some data for that. But I think ultimately for cardiovascular purposes as well. This is one, so just to give you an example of something that we've made for one of our folks in the Texas Medical Center in Baylor. There's a wonderful tropical medicine department run by Peter Hotez, and they've been studying Chagas disease, which, as you know, is the major cause of heart failure in South America. So this is an infectious disease that causes heart failure in South America. Unfortunately, with global warming, the parasite that carries it, the assassin bug, is moving north, and it's now being reported in Texas and southwestern states as well. The parasite that it carries, Trypanosoma cruzi, invades the bloodstream, localizes in the heart, causes inflammation that ultimately leads to heart failure. And the folks at Baylor, Peter Hotez's group, have developed a vaccine idea based on an antigen that they think that is exquisitely sensitive to immunization. TC24 is the protein. And we've made the RNA for them and put it into lipid nanoparticles, and it's working very well, clearing the Trypanosoma cruzi from the heart. I'll let them report that data out, but this is coming down the pike. This data has been already reported. William Pu and colleagues at Harvard have asked us to generate activated YAP for them. It's a transcriptional factor we've heard about already at this meeting. that's important in cardiac regeneration. And in their murine ischemia reperfusion model, they've shown that IM injections of the active YAP and message RNA reduces necrosis of the heart, scar, inflammation, and hypertrophy in their ischemia reperfusion model, associated with improved function. This is something that's, that we're doing at our place. You've heard Rod Pettigrew mention it this morning. I'll go into it a little bit more detail right now. This is human telomerase for age-related diseases, and it's beginning to look pretty interesting. We made iPSC endothelial cells and vascular smooth muscle cells from these children with progeria. And as Rod was telling you earlier this morning, these children have accelerated aging due to a lamin A mutation that induces progerin, which binds to the nuclear membrane to alter the nuclear membrane and alter the morphology of the nucleus and alter transcriptional profile. These kids manifest a senescence-associated secretory phenotype and accelerated aging that results in heart attack and stroke at the age, in the teen years, mid-teen years. So we also studied the mouse model of progeria, which we obtained from Francis Collins' mouse line. And essentially we asked the question, could we improve this condition? Could we improve this condition at the cellular level with human telomerase? Because associated with this disease, although the primary mutation is in lamin A, associated with this disease are short telomeres, as you can see here. So these are the parental HGPS endothelial cells. So the parents of these kids are normal. This is a spontaneous mutation. The children's telomere length is short. This is by Q-fish technology, so the short telomeres are observed. Now, we were able to increase the telomere length up to normal levels with just 2 transfections of the messenger RNA telomerase. So that was nice, but not totally unexpected. This is the proliferation using impedance accelerance equipment. And you can see the normal proliferation here in black, and the impaired proliferation of the endothelial cells, progeria IPS endothelial cells, and then after TERT treatment. So we were able to restore normal proliferation, cell replication. But there were some things we observed that we did not expect. One of those observations was, as was mentioned by Rod earlier, was the abnormal lobulated nuclei of the cells of these children was restored to normality. And not in all the cells, but a majority of the cells had normalization of the nuclear morphology. Another thing that we observed was that, and it's been observed previously, that in HGPS there's a lot of DNA damage occurring. With aging in general, there's a lot of DNA damage occurring. Oxidative stress and other factors are causing DNA damage, and that's shown here using a marker, 53BP1 and gamma H2AX. Both of those were increased in the nucleus of the cells from these kids. But the telomerase treatment reversed the DNA damage. This is genomic DNA damage reversed by telomerase. Now, we don't understand the mechanisms yet of these non-canonical effects of telomerase, but telomerase seems to do more than just extend a telomere. It reduces DNA damage. That's actually been reported about 10 years ago and forgotten. And we're now wanting to understand the mechanisms of this, and also the striking improvement in nuclear morphology is associated with a normalization of the transcriptional profile. We picked out the genes here that are differentially regulated in progeria. You can see there's a lot of them that are downregulated or upregulated. Many of the upregulated genes are those that encode senescence and associated secretory phenotype genes, inflammatory cytokines. And you can see with 2 transfections of RNA telomerase, it wasn't totally normalized, but it was improved, the transcriptional profile. I want to say one thing about this more. We also showed, I don't have the data here, but we were able to treat the animals, the mice, with telomerase as well. But in this case, because we hadn't yet developed a method for delivering the RNA telomerase with lipid nanoparticles that could be tolerated by the mice. We used a lentiviral vector, and that had many of the same effects in the mice. Lentiviral vector encoding telomerase had many of the same effects, beneficial effects on the mice, with a restoration of a reversal of vascular aging and vascular DNA damage. And the animals lived longer. Now, I want to say one thing. We haven't published yet, but we are going to publish it. When we made lipid nanoparticles with the mRNA telomerase and infused that into the animals, we were expecting to see an increase in longevity. We didn't. The animals died after about 4 months of infusions. And we learned that the lipid nanoparticles are toxic when infused systemically. We got hepatotoxicity that is something that we're going to have to overcome. And I think the whole field is going to have to overcome these problems with the lipid nanoparticles, which are great when you're using them as a vaccine, but they have some inflammatory activity themselves. And that causes problems when they're infused systemically for long periods of time. So that is a problem we're going to have to fix. But in the meantime, we've started to look into topical delivery of the telomerase, because we can avoid the problem with systemic delivery. We're working with a company, Avita, that has this product for burn patients. It's a spray-on skin. They take the skin from a non-injured area and disaggregate it, and then spray that on the burn site. And it works very well in young folks, not so well in older folks. So what they asked us to do is to add the H-TERT. So we put the H-TERT into lipid nanoparticles. We had an experiment with the lipid nanoparticles and found that the ionizable lipid nanoparticles don't work very well with skin, but the cationic lipid nanoparticles work much better. And we used this mouse model of wounding to investigate the benefits on the human skin cells of the telomerase transfection. We did one telomerase transfection of the disaggregated skin cells and placed them on the mouse wound. And what we looked at initially was we used a human lamin just to count the cells on the mouse. Ki67 is a proliferation marker, P21 is a senescence marker, and 53BP1 is a DNA damage marker. And what we found was that we did increase proliferation of the cells. We had more human cells after 7 days. We reduced senescence markers, and we reduced DNA damage. The company was encouraged enough by that we're going into a large animal model, going into pig model, because that's much more similar to human skin. I mentioned the off-target effects of lipid nanoparticles that are going to have to be overcome for systemic infusions. We're working with Nanovation, founded by Peter Cullis, who's a giant in the field of lipid nanoparticles, been working there for 50 years, and he's got some non-toxic lipid nanoparticles that we're going to be using systemically. Delivery to the tissue of choice is an issue. Talk about how to address that. Preferential tissue expression, and the lability of the messenger RNA. Messenger RNA doesn't last very long. It lasts hours to, well, minutes to hours, really, maybe a day, but depending upon the messenger RNA. But there's some fixes for that, which we'll talk about in just a moment. We published this paper in 2020 about a method for targeting inflamed tissues. What we did with our nanomedicine colleagues is to embed in the lipid nanoparticles membrane proteins from white blood cells. And it was really interesting that we called these leukosomes. The leukosomes ended up migrating to sites of inflammation. In a model of hypercholesterolemia, ApoE deficient mouse, we found that the leukosomes had a much greater preference for the vessel wall than did the normal liposomes. And we were able to deliver, in this case, rapamycin to the aorta of the ApoE mice, and we found that that reduced macrophage proliferation, reduced vascular inflammation. But the group at Penn, John Epstein, has taken this much further. What they've done is to specifically label lipid nanoparticles with anti-CD5, and you may have seen this paper in Science last year. Really incredible piece of work where they directed the lipid nanoparticles to target T cells. And what they're trying to do here is to generate chimeric antigen receptor T cells in vivo. And that, if that can be done, that would be amazing, because right now the CAR T cells, although very useful for fluid tumors, is not, it's very difficult because you have to take the T cells out, you have to expand them, and it makes things difficult. But he can target the T cells directly with the anti-CD5 and delivered a chimeric antigen receptor encoded by messenger RNA against activated fibroblasts to reduce cardiac fibrosis. We heard about this yesterday from Jay Zhang, who's using this model of RNA to use the microRNA library of cells to target them with RNA. So in this case, you have two RNA constructs. One encodes a suppressor protein, L7AE, that binds to this K motif on your other RNA, encoding the gene of choice, and blocks its translation everywhere except those cells that have microRNA that are encoded in the 3' UTR of the repressor protein. And so he was able to, Lior Zhangyi was able to show specific, cardiomyocyte-specific expression, in this case of GFP, but later went on to express VEGF or other proteins with this specific RNA system. I'm going to skip over this one because of time. Self-amplifying RNA, I'll just mention briefly. Basically, it's like linear RNA because it encodes the same antigen that you're interested in, 5' UTR, 3' UTR, your antigen of choice. But with the self-amplifying RNA, you do have a long segment that encodes a replicase that will replicate the RNA so you get more copies. The advantage of self-amplifying RNA is that it makes a lot more RNA for the same amount of molecules of the therapeutic RNA. You get much more RNA. It gets produced for a longer period of time. The problem is it's a very large molecule, 10 kb, and that causes some changes in manufacturing and purification. It also is somewhat antigenic itself. Circular RNA, I think, is a better approach. Circular RNA with an iris allows you to generate proteins so you can have your gene of choice in this circular RNA. You need an iris, an internal ribosome entry site for the translational machinery to grab hold of the circle and generate your protein of choice. The advantage is that it doesn't have 3' and 5' end. It's less susceptible to exonucleases. It lasts longer, up to 10 days we've shown in our hands. And then finally, to close, I'm going to mention this idea of personalized cardiovascular therapy. Callum McRae received $70 million from the American Heart Association for his one grand idea that we need to phenotype more deeply coronary artery disease, and he's done so with a lot of omics and imaging to computationally define many different classes of coronary artery disease. But the problem is, how do we treat these individuals? And I think small molecule therapies are not going to make it. We have to use genetic therapies that we can generate quickly for personalized therapeutics. And that is already happening in the cancer field, as you know. This paper just a couple weeks ago in Nature showed that personalized RNA neoantigen vaccines can actually work in patients with pancreatic cancer, which is amazing because that's a terrible, deadly disease. These constructs contain 20 neoantigens derived from the transcriptional profiling of the tumor and an algorithm that gave the investigators the immunogenic differences between the cancer and the normal tissue. And the responders in red did very well. The responders were classified as having a T-cell immunoreactivity by ELISpot, and the non-responders did not do very well. So an immune response generated against pancreatic cancer by an RNA vaccine seems possible. So to conclude, the clinical applications for RNA therapeutics are nearly limitless. Applications will be broadened by RNA innovations to increase stability, translational efficiency, and preferential tissue expression. And tissue and organ-specific delivery is coming. And then I do believe 5 years from now we'll have regional centers of RNA therapeutics throughout the country with these boxes I was showing you earlier. And we're going to have a democratization of RNA therapeutics. We're going to have RNA therapeutics delivered at the hospital. Thank you for your attention.
Excellent talk. You said today that when you introduce the telomerase, you see a rescue in gene expression. But my question is this rescue associated to the improvement of the nuclear shape that you guys see?
I didn't hear the last part.
So I want to know if the rescue of gene expression is related to the improvement in the shape of the nucleus that you see.
Possibly. That's possibly true. I didn't have time to show all the data. We have a marked improvement in functions. We looked at many different endothelial functions, nitric oxide release, and ability to generate tubes in matrigel, that kind of thing. The mechanisms are not completely understood, but when you have a shortened telomere, it does induce a DNA damage response that increases oxidative stress, and we do know that oxidative stress impairs a lot of endothelial functions.
And sorry, the last question. So, and the improvement in the shape of the nucleus, why do you guys see that? Because these cells have the laminin A is mutated, right?
Yes.
So that's why you have this weird shape.
Yeah.
So why do you see an improvement?
We don't completely understand, but one of the things we did notice is that there was a reduction in progerin levels in the cells that were treated with TERT. So it could be simply just a reduction in progerin levels that was responsible for this effect. But I just think there's something more to it, and we're trying to understand that.
Okay, thank you.
Your usual strong talk, John. The observation of what's happening with developing mRNA-based vaccines for pancreatic cancer is a tremendous welcome, as we all know, that it is such a devastating disease, and to see this emerging is very hopeful. But my question pertains to the work with Avita. You mentioned that this works well in the young, but not in older persons and the more elderly. Do we understand fundamentally the reason for that?
You're talking about the impaired response?
Yeah, with the skin cell regeneration with Avita, the work you're doing with Avita.
Right. Well, I think senescence in general does impair cell replication, and the inflammatory cytokines that are made as part of that senescence-associated secretory phenotype impair a lot of cell functions, including neighboring cells that are not yet senescent. So I think what we're doing in this case is we're reducing the number of senescent cells. We're allowing them to get back into cell cycle. We're improving function of the cells. So I think that we're going to continue to study this. It looks promising. But I think we'll probably switch to circular RNA for this indication. I think we'll get a much better effect with the circular RNA with more prolonged expression. Yes, Thomas?
Thanks for a great overview of this really exciting field. Your vision with the box being in every hospital, I kind of share, but I wonder, how do you envision the regulatory aspect of this
Great question. Great
because
question.
BioNTech, for example, invested enormous effort to make the process approved, but not the product. So do you think that will be common standard in?
There'll need to be some regulatory reforms, and I think we'll need people like Victor Zhao to help us with this issue, but there's already been some movement. I don't know if you know the name Timothy Yu. He's at Harvard. He's created this N of 1 consortium. His interest is in small RNA. He works with ASOs, and he treated a single patient. He developed an ASO for a patient with Batten syndrome and delivered that ASO to the patient. The FDA allowed him to do this, go all the way from cell culture studies directly to human without any intervening animal studies because that particular construct was very similar to nusinersen, which had already been approved. And Janet Woodcock wrote an editorial accompanying his paper in the New England Journal of Medicine that discussed this issue of needing some regulatory reform to deal with genetic therapies for personalized therapy.
Yeah, thanks.
Thank you, John, again. Our next speaker will be Dr. Youyang Zhao. And Dr. Zhao is a William Schwarzschild Distinguished Research Professor, head for the section of Injury Repair and Regeneration Research and from Lurie Children's Hospital of Chicago. And he is also a faculty from Northwestern University School of Medicine. And the title of his talk today will be Reactivation of Aging-Impaired Endothelial Regeneration for Vascular Repair and Vascular Diseases, Nanoparticle-Directed Precision Gene Therapy.
Thank you. So first, my disclosure, I'm the founder of Chief Scientific Officer of the company. Mountain Therapeutics. This really try to address John's question about the delivery for this company. So here we'll talk about endothelial cell, more talk about pulmonary vascular endothelial cell and in the disease, ARDS. But the principle, the technology can apply to cardiovascular treatment and heart regeneration later on. So endothelial injury can be induced by endotoxin, risk factor, mechanic, and that can cause disruption of the junction or endothelial cell death. In terms of cell death, it can be apoptosis, necroptosis, pyroptosis, and after injury, there's a process in the regenerate endothelial cell. So the question is, what's the mechanism for regeneration? Where the new cell come from? To answer this question, we use a genetic lineage trace in mice and challenge the mice with sepsis, for example, IP lipopolysaccharide, LPS. So at 48 hours after LPS challenge, the pulmonary vascular endothelial population from 40% decreased to 25%, because we know this is the inflammation, severe inflammation causes endothelial death. And then we, at 84 and 5 days later, collect the and look at the pre-labeled GFP endothelial population. We found out in this young adult mice, 3-month-old mice, the pre-labeled endothelial population can repopulate, regenerate, and at 5 days reach 50% from 25%. So that means the young adult mice, the endothelial cell, has intrinsic regeneration program. And several years ago, actually more than now, it's more than 17 years ago, we published a paper. There's a transcription factor called FOXM1, Focal-head transcription factor M1. And this transcription factor is interesting. The expression is not induced, the expression is lower at the basal and not induced during the injury phase, but is induced at the 48 hour or even peak at the 72, 96 hours after sepsis challenge. So the gene is early induced in the repair phase, not induced in the injury phase. Therefore, we made the endothelial specific knockout for this transcription factor, as shown in figure B. This knockout mice has a similar nickness, the vascular nickness to liquid at 6 hours after sepsis challenge. That means the gene knockout has nothing to do with the injury. But at 48 and 96 hours, the white type mice can fully recover, so the vascular nickness go back to normal, and the endothelial specific knockout for FOXM1 has a persistent nicking. And this mice, if we use lethal dose LPS, they have a much higher mortality, and this mice actually die of pulmonary edema. So when we collect the non-tissue before the mice die, you can see the weight-to-jar ratio is that pulmonary edema is about 9, and normal mice is only 5 or something. And also by electronic scanning EM, you can see the microvascular is nicking. And we also have data show there's impaired endothelial proliferation in these mice. For vascular repair, besides endothelial regeneration, they also need to reform the junctions, therefore form the endothelial barrier. So we also want to see if FOXM1 has anything to do with junction reannealing. In that case, we use another model, a quick junction disruption model. So treat the mice with thrombin agonist, and it can disrupt endothelial barrier function as quick as 1 hour, 30 minutes, 1 hour or 30 minutes in vivo. So in the wild type mice and the knockout mice, in figure A, you can see they have similar likeness at like 30 minutes after thrombin agonist peptide treatment. And the wild type mice can fully recover with 3 hours. The endothelial specific knockout cannot recover in 4 or 5 hours. In cell culture study, we found that this just goes through FOXM1, regular beta-catenin. And the beta-catenin is a adherent junction protein, therefore, and that regulates the junction reannealing after injury. So in this data, we show FOXM1, a transcription factor, is induced after injury due to the repair phase, important for endothelial regeneration and also for endothelial reform adherent junction for vascular repair. So it's so important, what's the upstream signaling to maintain the FOXM1 induction after injury? To address this question, we treat the wild type mice with a different kinase inhibitor, like MAP kinase, PKC kinase, PR3 kinase, and Turned out only the PI3 kinase inhibitor worked mainly. At 24 hours after LPS challenge, that means treated the inhibitor after injury. And then look at the FOXMA expression. So at 72 hours, FOXMA expression is pretty much inhibitor and by messenger RNA, by protein level. And there's PI3 kinase, there's two classes. One is pioneer gamma, is GPCR dependent. Another is pioneer alpha, beta, delta. There's many receptor tyrosine kinase dependent isoform. So therefore, we treat the mice with pioneer gamma inhibitor, AS compound, or pioneer alpha inhibitor, PI compound. And it turned out only pioneer gamma inhibitor can block FOXMA expression. And then the pioneer gamma knockout do inhibit the FOXMA induction after LPS challenge. And in that case, we want to see if the pioneer gamma knockout mice has a similar defect in vascular repair, and then overexpress FOXM1 in the pioneer gamma knockout mice can restore the vascular repair. So as you can see, the same. Within 24 hours after LPS challenge, the wild type of mice, pioneer gamma knockout, FOXM transgenic mice has the same injury. Vascular weakness is the same. And then within 72 hours, the wild type of mice is fully recovered. The pioneer gamma knockout is not recovered, just like the FOXM1 endosomal knockout. They are persistent leaking. The pioneer gamma knockout with the FOXM1 transgenic overexpress, they has same phenotype like a wild type. They can fully recover. And by scanning EM, you can see the same thing. So far I'm talking about the permeable vascular. Does this single pathway has a similar function in the systemic vasculature? So we use a two-photon microscopy to look at the cranial muscle venue. In that case, in the basal, we know the vascular is normal. So pioneer gamma knockout with FOXM1 transgenic, with the fixed texture, they're all no leaking. And then at 48 hours post-LPS challenge, the wild type of mice is pretty much recovered. So the fixed texture is in the vasculature. And the pioneer gamma knockout, because it's leaking, that's why you see the texture is outside of the vessel. With the FOXM1 transgenic mice that has normal repair. So this data indicate the pioneer gamma FOXM1 pathway is not unique to the permeable vascular and the sentinel cell. Is also in the systemic vascular for endothelial regeneration vascular repair. In the ARDS patient sample, we look at the pioneer gamma expression. In normal sample, you can see pioneer gamma is highly expressed in the vascular endothelial cell, but in non-survivor ARDS patient, and actually the pioneer gamma expression is diminished. So this is the kind of mimic what we see in the mice. So we just talk about pioneer gamma is a GPCR dependent PI3 kinase. So there's more than 200 GPCR expressed in human vascular endothelial cell. With the GPCR, activate pioneer gamma is a bigger question for endothelial regeneration. To look at 200, you must have a powerful, efficient method to modify gene expression in vascular endothelial cell. AAV, the virus lipid nanoparticle, cannot tackle endothelial for gene delivery. After IV injection, all this go to the liver. Now for this tackle vascular endothelial, just like Cooke talked about, that's before my paper, pretty much that we don't have a method to tackle vascular endothelial after IV injection. So we took a fellow in my lab. She was trained in nanomedicine. So we did a nanoparticle screening. This is a polymer nanoparticle. We found a formulation of nanoparticle if you do IV injection, as I show here, in the mice, has a whole body distribution. It's not accumulated into the liver. So this makes us to further modify, make it can carry DNA or mRNA. Anyway, the nanoparticle is positively charged, so it can deliver nucleic acid. Here, we mix the nanoparticle with the plasmid DNA, express the CRISPR system, Cas9, and also the guanine in one plasmid. And after 10 minutes in room temperature, and they do IV injection to the mice. After 5 or 7 days, collect the tissue to look at the gene in vivo endothelial CRISPR genome editing. As shown in the in figure C, after 5 days, pioneer gamma expression is pretty much totally diminished, just like a pioneer gamma knockout. And the protein level is about 80% deficient in the endothelial cell, but in the other cell type is not affected. And the phenotype, just like what I talked about in the pioneer gamma knockout, with guard RNA in the plasmid. Pionate gamma is diminished. They have persistent leaking at 72 hours after sepsis. FOXP1 expression is inhibited. That's in the lung. How about in the heart and the aorta and the other vasculature? So we look at the heart. Similar to the lung, about 80% of protein knockdown in the heart, vascular endothelial cell. In the aorta, you can see from figure C, also very efficient. Pionate gamma expression is totally diminished. And then even in the peripheral vessel, in the macrovascular endothelial cell, pionate gamma expression is diminished. Another advantage for this approach is because of the plasmid DNA, there's no limitation in size. So we try to figure out, can we do 2 gene knockout at the same time overexpress gene? So we made a construct, put 2 different guard RNA. One is the pionate gamma, one is the VEGFR2. Target 2 gene, knockout 2 gene. So we look at the genomic editing efficiency. Single one, about 40%, and the double one, the same. So you can make a double knockout in 1 injection. At the same time, we express GFP. So you can make a 3 gene modification, knockout 2 gene, overexpress 1 gene in 1 injection, no problem. Another construct, 50 kb, what we did is knockout 1 gene, pionate gamma. At the same time, overexpress 2 gene, one is the GFP, one is the FOXP1. So this is like the transgenic knockout, pionate gamma knockout, overexpress FOXP1. Here, we do this in 1 week. So inject the plasmid DNA, express the guard RNA for pionate gamma, and also express FOXP1. You do not need to breed the mice for 2 years. Here, as we show here, this plasmid can reinduce, because FOXP1 expression can reinduce FOXP1 expression in the vascular endothelial cell, and the pionate gamma guard RNA knockdown suppress FOXP1 induction. And the vascular repair is impaired in the pionate gamma guard RNA plasmid DNA transduced mice, but with the plasmid DNA express FOXP1 is normalized. So basically, mimic the transgenic mice. Knockout the pionate gamma by guard RNA has impaired vascular repair, but if the plasmid DNA can express FOXP1 at the same time, they can normalize vascular repair. So now, let's go back to the question. What's the upstream GPCR, active pionate gamma PI3 kinase? So we look at the mouse vascular endothelial cell. We pick out the top 10 GPCR. We identify potent guard RNA for each 10 GPCR. Here, what I show is the 2 GPCR. One is the CXCR4, one is the S1PR1. And in figure B, A and B, we show both of this guard RNA is very efficient to knock out the CXCR4 in the vascular endothelial cell or in the pionate gamma, in the S1PR1. Depends on which guard RNA using in the plasmid. And then actually we gave this plasmid DNA to the GFP-enabled endothelial cell mice. And then we look at the GFP endothelial population at 96 hours post-sepsis challenge. In the S1PR1 guard RNA mice, the GFP population go back to totally normal. That means S1PR1 is not involved in endothelial regeneration vascular repair. CXCR4 guard RNA plasmid transduced mice has impaired recovery of GFP endothelial. So XCR4 is immediate endothelial cell regeneration, and the total endothelial cell in the lung is not recovered, is about 40%. The white have normal control about 50%, and the S1P1 guard RNA is fully recovered. So this really demonstrates CXCR4, the GPCR is a mediated P10 gamma PI3 kinase activation for regeneration. Because we talk about endothelial proliferation regeneration, everybody may be easy to think about VEGFR2, VEGF signaling. So we also made a VEGFR2 guard RNA plasmid DNA delivered by the nanoparticle, and after 5 days, we challenged with the, we collect the non-tissue, look at the VEGFR2 expression. It's more than 75% of protein knockdown by this plasmid DNA nanoparticle administration. And then we challenged the mice with sepsis and collected 4 days. The GFP endothelial population is totally full recovered, just like the control mice, and the total endothelial population is full recovered. This demonstrates VEGFR2 signaling, the normal angiogenesis required signaling, is not involved in endosomal regeneration, specifically at least in this sepsis inflammatory injury model. So then we use this guard-RNA plasmid DNA system and challenge the mice with sepsis, look at the vascular repair mechanism. So we gave a guard-RNA 1 for CXF4. That's the efficient guard-RNA. Guard-RNA 2 has no efficiency. It's a poor guard-RNA. And as we show here, use a guard-RNA plasmid DNA nanoparticle delivery at 72 hours. They have impaired vascular repair, just like the CX4 genetic endosomal knockout mice. And then this mice also has a persistent inflammation indicated by mild peroxide activity. And again, by nanoparticle guard-RNA plasmid delivery, we show the same thing. So 1.5 year to 2 years breeding, you can do this in less than 1 month. So far what I show is we identify a single pathway. After sepsis challenge, SDF1, I didn't show the data here, that's a ligand for CXF4 is induced in the endosomal cell, and that activates the receptor CXF4, therefore leading to GPCR-dependent PR3 pionate gamma PR3 kinase activation, not the VEGF2 activation, and that mediates FOXP1 transcription factor induction, therefore leading to endosomal proliferation and regeneration, and also for beta-catenin induction, and that leading to the new endosomal cell, existing endosomal cell, or new endosomal formed at the current junction and for vascular repair, therefore resolve inflammation. As we talk about, there's a question about aging. We see that in pneumonia, ARDS, COVID-19 patients, there's a high incidence of sepsis and ARDS, and the mortality is much high in elderly patients. So what's the mechanism of that? We use this aged mice with genetic labeled endosomal cell, challenged with sepsis, and they look at the endosomal regeneration. So in the young mice I just showed before, and also here again in figure C, we know within 5 days the endosomal cell can fully recover in the 3-year-old mice. From 40% go down to 25%, and within 5 days, they fully recover to 40%. In the aged 20-month-old mice, endosomal cell is also about 40% in the lung, and they go down to 20%, similar injury. But after 5 days, there's no endosomal regeneration. The endosomal population is still only 20%. So we think this is a mechanism for the high mortality because they impair the vascular repair. Here we show in the aged mice there's impaired endosomal proliferation compared to the young adult mice, and the vascular permeability in the young adult mice within 96 hours post-LPS challenge is fully recovered, just like a basal, but the aged mice is persistent leaking. Inflammation is the same. And we look at the FOXP1 expression because we know FOXP1 endosomal cell after injury is induced really for endosomal regeneration. So here in the aged mice, FOXP1 is not induced, but in the young adult mice, FOXP1 is induced. So we use this nanoparticle to deliver just one gene, FOXP1, to the aged mice, 22, 24 months old. At 24 hours post-LPS challenge, we mimic clinical situation. And want to see if just overexpress one gene after injury, can you restore endosomal regeneration in the aged mice? So we look at the Bioengineering blood, of course, we can also do in staining to show FOXP1 expression can be overexpressed by nanoparticle delivery. And then we look at endosomal proliferation in this nanoparticle delivered FOXP1 overexpressed mice. They had significant increase in endosomal proliferation in the aged mice, and that also leading to the cell cycle gene downstream of FOXP1 target, like CDC25C, cyclin A2B1, and then we look at the vascular permeability. So if the mice in the endosomal overexpressed FOXP1, they have normal vascular repair, and with the vector, they are persistent leaking in the aged mice, and the inflammation is persistent high in the vector mice, but not in the FOXP1 overexpressed mice. So this is, we use a CDH5 promoter in the plasmid DNA, express FOXP1 in all endosomal cells. because this is plasmid. So we want to see if we just overexpress FOXM1 in the pulmonary vascular endothelial cell, can you promote vascular regeneration in the pulmonary vascular endothelial cell? In this case, in the plasmid, we use two promoters. One is the pan-CDH5 promoter. Another one is TMEM100. This has to be shown recently. TMEM100 also by Dr. Joseph Wu's paper. TMEM100 is pretty much a gene highly expressed in the pulmonary vascular endothelial cell, not in the heart, not in the brain, but it also has a little bit expression in other cell types like atrial cardiomyocytes. That's why we use two promoters to limit the gene only in the pulmonary vascular endothelial cell. In that case, we use the nanoparticle deliver the plasmid DNA over FOXM1 only in the pulmonary vascular endothelial cell, as we can see here. In that case, has the same phenotype. With the vector, is a persistent leaking at 72 or 96 hours, but with FOXM1 earlier expressed in the pulmonary vascular endothelial cell, there's normal vascular repair, inflammation is resolved by minor peroxide activity and also interleukin 6. So this is the kind of procedure gene therapy in the future. If you use a vascular-based specific promoter, you can eliminate the gene expression or knockout in the pulmonary vascular endothelial cell or in the cardiac endothelial cell. We just talk about potential clinical relevance. So we look at the FOXM1 expression in the normal healthy human tissue. This is about age 50. So FOXM1 is by RNA scope in situ hybridization. FOXM1 is not expressed in normally. Otherwise, it may cause cancer. That's why FOXM1 is not expressed in normal cells. And in COVID-19 patient, in age of around the 50s, we look at the FOXM1. It's just like in young adult mice. FOXM1 is reinduced in the vascular endothelial cell in middle-aged COVID patient. But in this we look at the three COVID-19 patients in age 80s. Pretty much you don't see FOXM1 induction. So that's why the elderly COVID patient or pneumonia patient has a high instance of ARDS and high mortality because they don't have this endothelial regeneration mechanism. FOXM1 is not induced. So I think the next step now is to test our nanoparticle to see if it's safe, it can be effective in the natural animal model. If that's the case, maybe we can use this either mRNA technology or plasmid DNA, use the nanoparticle to IV injection, deliver them to elderly patient to see if we can normalize reactive vascular regeneration for vascular repair in the elderly patient to treat ARDS. Another thing what we did, because this is a plasmid DNA, you can do CRISPR system, you can do prime editing. So we found if you, in the aged mice, if you use a nanoparticle, deliver RNA CRISPR system, knock out the FOXO, that's another fork head transfer factor in 22 months old mice before LPS. What it means is maybe in an elderly patient, if we use the nanoparticle deliver gene, deliver RNA, knock out the FOXO, hopefully just like in the mice side, and they had maintain the endothelial regenerative potential. So in that case, elderly subjects after COVID, they can have the normal endothelial regeneration program just like a young middle-aged subject can have a full recovery. So I think this maybe is another really big thing we want to do to rejuvenate the endothelial cell to prevent or to maintain endothelial regenerative potential in the elderly subject to inhibit, to resistant to sepsis, pneumonia, COVID-induced injury and repair. So I will skip this one here because this is a cardiac regeneration. So I think the next question is really, can we use this nanoparticle, deliver modified RNA or plasmid DNA? If it's a plasmid DNA, even we can use a heart vascular endothelial specific promoter by only deliver the gene in the heart vascular endothelial cell, not in the aorta, and or not in the pulmonary vascular. So you can do really eliminate it specifically in the heart endothelial cell, and that can deliver this paracrine effect like Dr. Zhao Victor Zhao talked about yesterday. For example, SFRP2 or combo microRNA, or even cardiotrophin-1 or neuroendocrine agrin. Agrin has to be shown released from endosomal cell can promote cardiomyocyte regeneration in adult mice. And also, you can deliver any factor that can activate vascularization, and that's required for heart repair. So I think there's a bigger potential for this nanoparticle deliver gene, and even potentially in the cardiac endosomal specifically, there's less effective precision gene therapy for and cardiac repair, recovery, and cardiomyocyte regeneration potentially. So this is really the work for two members, lab members, Xiaoming and Xiaojia, and by collaboration with Ezra America from UIC, Roberto Costa, and also Kurt and Yuan from Fudan University, and also by NIH funding. Thank you.
Exciting data. Can you tell us a little bit more about the composition and biodistribution of your lipid nanoparticle? Is it a lipid nanoparticle, first of all?
It's a polymer nanoparticle. It's not lipid. Polymer nanoparticle. It's a PLGA modified with PEG. So this is published in the paper Cell Reports last January.
It's biodistribution?
Biodistribution is very nice. It's a whole body, and we did the time course 8 hours, 24 hours. So look like the pretty much degraded within 48 hours, but the plasmid DNA can maintain exposure as long as 7 days. So for gene overexpress, we need to do weekly injection. For CRISPR, 1 injection, that's it. And in terms of toxicity, it's pretty much in the mice we don't see toxicity. We did a 6 injection for my collaborator for aorta atherosclerosis study, do weekly injection for 6 times, overexpress, no problem.
Thank you, Dr. Zhao.
So the next speaker is Yang Zhou, assistant professor from UIB. Her talk, the title is Transcriptional and Epigenetic Reconfiguration in Direct Cardiac Reprogramming.
Thank you for the introduction, and I also wanted to thank Dr. Fukuda, Jay, and Yuji to invite me to this fantastic meeting, and it is my pleasure to have this opportunity. So I was trained in the basic molecular and cellular biology, and this is actually my favorite epigenetic one-kingdom model. So it is kind of like simply and nicely show the process of normal development from the pluripotent stem cells to the somatic cells. And then in the middle, it shows the process of the pluripotent reprogramming and follow the differentiation. And for the right one, showing you the direct conversion between different types of somatic cells. So the lower diagrams showed you that the cardioregenerative capacity decreased along the evolution and from the lower vertebrates to mammals. Recently, we also know that in mouse and the pigs, the heart regeneration is diminished shortly after birth. Very interestingly, we actually can modify the Wendling model to help us understand the relationship between the cardiomyocyte maturation and the regeneration. So now the ball on the top of the hill represents the proliferating cardiomyocytes. When it rolls down, it becomes the mature, non-proliferating cardiomyocytes. And this model also provides a summary, nice summary for the 2 strategies so far we have to generate new cardiomyocytes. One is to stimulate the stimulation of existing cardiomyocytes for the cell cycle reentry, which in that case, we will see we need to kind of like push back the board on the top and let it roll down again. And yes, this topic has been like extensively discussed yesterday. And the other one is direct conversion of cardiofibroblasts into cardiomyocytes. So please keep in mind this like ridges and valleys in this model. They are very important, and we have to overcome these hurdles to allow the successful self-weight conversion. So more importantly, these two strategies are very promising for the translational application because these two strategies can lead to the in situ heart repair by directly deliver genes, small moleculars, modifier RNAs, and microRNAs in the injured heart site to target different types of cardiac cells. So let's focus on the direct cardiac reprogramming. The pioneer works were done by Masaki Ida and Li Chen, who are here today, and they actually only transduced the 3 factors in mouse fibroblast, and after 2 weeks, they observed the functional cardiomyocytes. This conversion can also be done in the injured mouse model in vivo, and after 4 weeks of directly injection of the virus, you will see the reduced scar size and the restored heart function. So here's some advantages of this technology. It is a simple and quick process to achieve in situ cardiomyocyte regeneration. It has dual benefits to generate new cardiomyocyte and reduce the fibrosis. It also bypasses the progenitor stage and minimizes the tumorigenesis risk. However, when we adapted this approach into human cells, we faced several challenges. We actually need additional factors. We observed the slow process, low efficiency, and no contractility. So in 2019, we reported a new cocktail that can be used for human direct cardiac reprogramming. It includes MAF2C, GARDA4, TBX5, and the microRNA 133. And for short, we call it MGT133. So utilizing this platform, we try to understand why the human direct cardiac reprogramming is limited. So we just performed time course single-cell RNA sequencing analysis. We actually selected time points based on the expression of cocktail MAF2C and the cardiac troponin T expression. And the samples were applied to the single-cell capture by C1 fluidized system and the next generation sequencing. To make a long story short, we actually reconstructed the trajectory of the cell fate changes after the viral transduction, and we find a bifurcated pathway that either go to the reprogramming route or just go back to the fibroblast identity. Also, we want to compare our human data with the mouse data we generated. So we developed a cell-fate index, which can be used to evaluate the reprogramming progression in 2 species. So here you can see that the index of human reprogramming cells is lower than that in mouse, especially for those genes related to cardiac structure, cell junction, contractility, and ion channels. So our data, all our data suggests that the incomplete transition of cardiomyocyte fate. So if you remember the one in the model I mentioned, and there are like ridges and valleys there. So we were trying to identify what are those. Then to obtain the complete cardiomyocyte identity, we wanted to find a way to activate essential cardiac function related genes. So we hypothesized that we may need additional epigenetic or transcription regulators, but these are not only like for the factors that for the ridges or valleys. There are possibly other like metabolism regulated genes also, but we focus on the epigenetic and the transcription regulators. So firstly, we identify the important epigenetic regulators. We focus on the well-known repressive histone mark, H3K27 trimethylation, which has been discussed by Victor Zhao yesterday. And we have the global profiling of H3K27 trimethylation in our MGT133-induced reprogrammed cardiomyocytes. And very surprisingly, we saw there are still abundant occupancy of H3K27 trimethylation on the cardiac gene bodies. And we indeed, through a loss of function screen to identify a potential barrier, EZH2, that might be associated with H3K27 trimethylation. The knockdown of EZH2 can enhance the reprogramming efficiency. And here is the gene expression analysis showing you that after EZH2 knockdown, we saw the gene program related to muscle contraction, myofiber assembly increased. All the downregulated genes are associated with mitotic cell cycle and cell proliferation. So EZH2 is a catalytic unit of polycomb repressive complex 2, PRC2. It has a set domain which can remove the methyl transfer to the histone lysine 27 residue. So not only through the genetic knockdown approach, we can also use the pharmacological inhibition to show the enhanced reprogramming. And here we also found that the genes de-repressed by the EZH2 inhibition harbor the more abundant H3K27 signals. Then next, we also wanted to identify the missing key transcription regulators that can activate additional cardiac function-related genes. To do that, we wanted to identify the underrepresented genes compared to the reprogrammed cardiomyocyte and the endogenous functional cardiomyocytes. So we firstly determined more than like 200 cardiomyocyte-specific transcription and epigenetic genes to compare the fibroblast with the functional cardiomyocytes. And then we rank these genes based on their expression in reprogrammed cardiomyocytes. And it is clearly you can see that here that some of them, like MAF2C, TBX5, and GATA4, has been successfully activated. However, some of the genes are merely activated, but they are very important and highly expressed in the functional cardiomyocyte. TBX20 is one of the top missed factors we identified here. So this qPCR data showed you that the TBX20 expression is very low in the fibroblast, but high, very high in the embryonic stem cell derived cardiomyocytes. Our current reprogramming cocktail, MGT133, is unable to activate this gene. So then we overexpressed TBX20 and demonstrated that it can enhance the quality and the quantity of human-induced cardiomyocytes. And here's the immunostaining showing you the enhanced expression of the maturation marker, cardiac troponin I, alpha-actinin, and also the global gene expression showed that the activated gene programs related to cell junction, action potential, and sarcomere that are very important for the cardiomyocyte maturation. And this confocal image nicely showed that the induced cardiomyocyte with TBX20 transduced has much better alignment of the sarcomere structure. More importantly, TBX20 can also lead to the functional maturation in human-induced ICMs. So we can record the strong signals of calcium flux in TBX20 group, which is very similar to the iPS-derived cardiomyocyte, and find more beating cells in TBX20 group. After the co-culture, we can also capture the action potential in TBX20 group only, not any in the control group, control EV groups. We also know that mitochondrial function is very important for the cardiomyocyte maturation, and it provides the energy resource for the contractility. So we find that TBX20 improves the mitochondrial functions by evaluating the mitochondrial numbers and the mass by mitochondrial tracker and the mitochondrial DNA evaluation. We also measure the ATP production in different groups by seahorse and find TBX20 promote the switch from the glycolysis to the fatty acid oxidation, highlighting the impact of TBX20 on the mitochondrial function. And also there's an important question we always be asked, that is how much like a human-induced cardiomyocyte resemble endogenous cardiomyocyte. To answer this question, we performed a single-cell RNA sequencing analysis, and then we mapped it to the reference human cell atlas, which has all the like cardiac cells population, and then through Symphony, we have this map showing you the human cell atlas, adult and the ventricular cardiomyocyte. And then we plot our data to this reference map, and we find that more TBX20 cells are showing the ventricular cardiomyocyte features, suggesting that TBX20 may also help to switch the ventricular and atrial cardiomyocyte identity after reprogramming. So, so far I've already showed you that TBX20 might be a maturation boosters during their cardiac reprogramming in human cells. It leads to the improved sarcomere contractility and the metabolism. But the question is how TBX20 coordinate with current reprogramming factors, the MGT, to activate the downstream cardiac genes. So to answer this question, we performed multiple DNA profilings of, for the TBX20 and the histone marks. Here we show that only in the TBX20 group, we see the increased chromatin accessibility and increased enhancer activity showed by the H3K27 acylation density plot. And also utilizing the human tissue enhancer database, we find that TBX20 peaks are mostly enriched in heart tissue. And here's the example showed you that only with TBX20 bind, which is indicated by the red bar, we have increased the H3K27 acylation, increased the ATAC signal, and decreased the repressive H3K27 translation. So we also investigated the relationship between TBX20 and the current MGT factors. And this is the density plot showed you that with TBX20, we will see much higher binding regions, more binding regions of MAF2C, GALA4, and the TBX5. Also, this is a histogram showed you like not only the one factor binding events, we also showed you the orange and the red color are the 2 factor and the 3 factor binding events. So TBX20 can have the synergetic effect that can promote the multiple binding events during this process. Also, using the luciferase reporter assay, we successfully demonstrate that synergetic effect, because here when we use TBX20 only, we can only see the moderate activation of the enhancer activity on like genes MYBPC3, but with MGT addition, we can see much higher activation of this enhancer activity. So with that, I just have a very simple summary that showed you that TBX20 can improve the quantity and quality of their cardiac reprogramming in human cells, and it functions through the coordination with MGT, bind on the cardiac enhancers and activated the whole cardiac gene programs. And if we call back to the Huntington model, we kind of like answer the question, what are those regions and the valleys that the understanding of those can help us to improve this process. So with that, I would like to thank all my lab members and my collaborators, the support from all the funding resources. Thank you so much for your attention, and I'm happy to take any question you may have.
Can I ask? Thank you very much for your great talk. I'm curious about the single cell data. How can you control the phenotype of ICMs ventricular type or atrial type. Does TBX20 affect this phenotype?
So TBX20, actually we checked the TBX20 expression in the Human Heart Atlas, which is a big database that includes all the human cardiac cell, single cell data in that. And we find the TBX20 is highly expressed in a very specialized subtype. They call it CM4, which show the very high kind of like mitochondrial or metabolic energy level. And for us, we just use an algorithm called Symphony, which we can use a reference map like the Human Heart Atlas I showed here, and then we can map our data on that, and to see our cell types, if they are more similar to the atrial or more similar to the ventricular. And we find that TBX20 infected cells are more similar to ventricular.
So more ventricular type?
Yeah.
Okay.
I know this is kind of like a little bit controversial to the current finding, because TBX20 during development we find TBX20 is important for the atrial development, but this is in kind of like a different context. So we think this is why it's important to investigate these factors in reprogramming context.
Thank you very much.
Thank you for your question.
Thank you, Dr.
All right, so now let's move on to our last speaker. And our last speaker is Dr. Saksvel Sadayapan. And he actually is the Vice Chair of Basic Research, Director of Heart Branch of the UC Heart, Lung, and Vascular Institute from University of Cincinnati College of Medicine. And the title of his talk is Cardiac Organoids for Hypertrophic Cardiomyopathy.
All right, so I want to start with thanking the organizers, Dr. Fugada and Dr. Zhang, for this wonderful opportunity to come and present our work. 20 minutes, long way coming from Cincinnati. Thank you so much. These are my disclosures. Oops. So many consultations and so on, but nothing to do with my today's presentation. So my lab has been interested in studying the disease called hypertrophic cardiomyopathy for a longer time from my PhD. It's a genetic heart disease, autosomal dominant, seriously 1 in 200 right now. But the problem is diastolic dysfunction, and patients have hypertrophic cardiomyopathy have very high number of sudden cardiac death, but also the clinical spectrum is highly heterogeneous. Hypertrophic cardiomyopathy is defined as the disease of the sarcomere because predominantly mutations, 80% are coming from sarcomeric genes, specifically myosin heavy chain and myosin binding protein C. My lab has been again studying on this particular protein, cardiac myosin binding protein C. It has more number of mutations than any other genes, and also representing over 40% of all mutations. And interestingly, 70% of the mutations are truncations. Coming to the disease mechanism, there are two major subtypes. One is haploinsufficiency. If you have one allele with the mutation, it's not enough to produce the full-length protein, whereas the second one is called poison polypeptide, where the mutant protein can cause poison effect. So in collaboration with Dr. Beth McNally and Roy Bucklewood, we have identified a gene mutation that is aspartic acid to valine in the codon 389V in myosin binding protein C as a polymorphic variant. Again, this is not in one family, it's not in one region, it's a polymorphic variant, almost like 5% of South Asian descendants coming from India, Pakistan, Bangladesh, and Sri Lanka, those regions. In patients, we were able to see that having this mutation, they show hypercontraction by increasing ejection fraction and fractional shortening. We published this paper in 2018, and we also in this study showed that using iPSC-derived cardiomyocytes, you can see they have increased in cell size, hypertrophic phenotype, in the iPSC-derived cardiomyocytes, but also they also showed irregular calcium transients, like a high number of cells. So in this paper, we kind of concluded that 389V is sufficient to cause hypercontractility in vitro. But again, coming to this theme of this seminar or this conference, I wanted to focus on what is the next level of model, in vitro model, to study this disease, like hypertrophic cardiomyopathy in vitro. So we have a lot of limitations in using iPSC-derived cardiomyocytes. It's a 2D model and lacking the 3D complex structure and also multicellular organization. And most importantly, 2D iPSC-derived cardiomyocytes, they do not have crosstalk between cell types. And also, still, the cell maturity is relatively moderate. And also, the heterogeneity in contractile function is still very weak. So there is a need for new model. So again, we know that this recent last 3-4 years, the theme of organoids is coming up. So many papers, they have now generated organoids for various organs. And one is cardiac organoids. And iPSC-derived cardiac organoids is the next model, still in the early development. So again, what are the advantages of using cardiac organoids? Well, it's a 3D model, number one. It can also include the in vivo complexity. We can add different cell types. For example, endothelials and fibroblast cells can be added into this co-culture to make the 3D organoids. And also crosstalk. Again, this can be modeled like an in vivo. So there are a lot of advantages in using or studying cardiac organoids in vitro. So for my own project, this is Darshini Desai, graduate student. She received the AHA American Heart Association pre-doctoral training grant to generate cardiac organoids for studying this mutation 389V in vitro. And so the objective was to establish and characterize the organoids in vitro and to define whether these organoids develop contractile dysfunctions, again, hypercontractility. So first 2 years, again, in a graduate student, sometimes, it's a challenging project to really develop this model. You can imagine, right? Many times she comes to my lab, my office, and sit and cry because there is no established protocol for this model. And so, but again, a lot of literature are coming now. And so she was able to develop finally an organoid. This is like a 20 days protocol. She was using the Wnt signaling activator and then inactivator and then B27 supplement with or without insulin. We use the agarose mold. So in one Petri plate, here you can see, we can have 36 organoids in one small mold you can generate. So this is a 20 to 28 day protocol. She was able to finally make it happen. Here is a beating organoid of 18 days old. You can see this is an individual organoid. It can beat like a heart. So using these organoids, we were able to characterize the gene mutation. First thing, we want to make sure organoids develop like cardiomyocytes or sarcomeres pattern in vitro. We were able to do the staining with alpha-actinin, and we were also doing a lot of Z-disc characterization of the sarcomeres. You can see on the right side the sarcomeric patterns using alpha-actinin staining. We also do the other staining of these organoids to make sure organoids are matured, they develop sarcomeric structures, and also closely to adult human heart. So, so here we have, again, these are pilot data. We have not published yet. Here is the whole mount cardiac organoid, as well as we have also done electron microscopic analysis to make sure there is a defined sarcomeres in these organoids. Then we were able to isolate RNA. We were able to make sure there is an equal amount of wild type and mutant allele in these organoids, and then also presence of the mutation in the mutant organoids. Next, we were able to use ion optic system, the cytomotion system, to measure contractility. Again, this is a very standard protocol. We can able to measure the peak, total time. It's include contractile velocity, and then amplitude, and then time to peak 50% and then 90%. You can see that very clearly these organoids with the 389V show hypercontraction or increased force in vitro. Also, we were able to measure calcium transients using flow 4, able to calculate the transient time and the ratio. Again, very clear that the calcium flux was also significantly increased in this 389V mutant organoids compared to the non-carriers. Then we were able to think like how this is matching with additional stress. For example, the GSH and GSS G ratio, it's a marker for oxidative stress. Again, we were able to see there is an increase significantly. And also using Seahorse experiment, we were able to see that the mitochondrial function is also enhanced in association with increased contractility. And lastly, we were able to do RNA-seq. Again, we were able to show the upregulation or activation of all the standard genes compared to non-carriers, including the calcium regulatory genes, cardiac contractile genes, as well as hypertrophic genes in these mutant organoids. So again, I think we were able to overall make sure that these organoids develop or show like a hypertrophic cardiomyopathy phenotype. So this is my summary of the slide. So we were able to generate cardiac organoids in vitro. The mutant is clearly showing the hypercontractility as well as increased oxidative stress. Again, we were able to see the increased calcium flux. And the ongoing experiments are, we want to make sure what are the cell types in these organoids by doing the spatial mapping, as well as using antisense oligos and mavacamten, which is one of the recently derived FDA-approved drug to treat hypertrophic cardiomyopathy. Now the question is, what are the challenges? Again, I'm not here to define this is the best model, but we are also undergoing a lot of challenges during this development of this organoid. Number one, batch-to-batch variability. Again, you saw our data, they are highly variable. And also, we have not included co-culture. For example, we have not used cardiac fibroblast or endothelial cells in this model, but this should be studied in the future, one of the future directions. Again, this lack of hormonal mediated stress and nutrient gradients, these are still kind of weakness in this model, but I'm hoping that in the coming years, this can be improved. With that, I want to acknowledge my lab members, specifically Darshini Desai. She's graduating next month, and she got a job already in BMS, again, continuing similar area of research. So, so with that, I want to acknowledge and thank my collaborators, Dr. Wang, Dr. Gu, and Dr. Krishna at University of Cincinnati and Cincinnati Children's Hospital. With that, I'm happy to take any question if you have time. Otherwise, we can meet at the lunch or in the evening at the reception. Thank you so much.
Thank you. Any question? Any question? I don't see anybody question. We can go to the lunch, and we want to. No, there is a question.
A short one. Thanks, Sakti. Nice talk. Our own experience with the organoids is that they have quite some variability. And I was wondering, why don't you use a much more established EHT system for measuring contractility? Because that's tested in 1,000 labs and it's just working.
Great question. Again, we look for some novelty in this.
Novelty, good.
Novelty, innovation, technology, yes.
No, honestly, I think it's important to consider what do we want to measure. If you want to measure developmental processes and maybe congenital something like that, I think for that, the organoids are really important.
Yeah.
But to measure plain contractility effects, I think a more simple model, which has much better defined manner, is the EHT.
I completely agree. I mean, ETH is well established, but again, we are looking unique and interesting model here, high throughput, right? If you want to put thousands of organoids, if they are consistent, this is the best model, then think of ETH. So EHT. Okay, guys, have a nice time in lunch.
Yeah,
Thank
let's
you.
give another round of applause to all speakers and of course audience. Yeah, now we can go to lunch.